In 2016, a team of scientists in Osaka, Japan, was digging through sediment behind a plastic bottle recycling plant. They weren't looking for anything special, but in that mud, they found a bacterium that shouldn't exist. By the end of this video, you'll know exactly how that bacterium's enzyme works, how scientists have since made it thousands of times more powerful using AI, and why the same plastic this enzyme destroys might already be sitting in your bloodstream right now. Let's rewind. Plastic bottles are made of something called PET, polyethylene terephthalate. It's basically a long chain of molecules linked together like a fence of interlocking rings, and that structure is exactly why plastic survives in the environment for centuries. Nothing in nature had ever evolved to break that fence apart. Until it did. The bacterium the researchers found, later named Ideonella sakaiensis, had developed two enzymes working as a team. The first, called PETase, latches onto the surface of the plastic and starts cutting those long chains into smaller pieces. The second enzyme, McGeehan, finishes the job, breaking those pieces down into simple building blocks, terephthalic acid and ethylene glycol. The bacterium then eats those building blocks for energy, the same way you digest sugar. In other words, this organism turned trash into lunch. Here's the part most videos skip, the actual mechanics. An enzyme is a protein folded into a very specific 3D shape, and that shape creates a pocket, an active site, that fits snugly around a target molecule. Think of it like a key built for one specific lock. PETase's active site is shaped to grip onto the surface of PET plastic. Once it locks on, it uses a chemical process called hydrolysis, essentially using a water molecule as a tiny pair of scissors to snip the bond holding the plastic chain together. Do that enough times, across enough of the surface, and a solid piece of plastic slowly dissolves from the outside in. Researchers at the University of Portsmouth and the U.S. Department of Energy's National Renewable Energy Laboratory mapped the crystal structure of PETase, atom by atom. And in the process of trying to understand it better, they accidentally engineered a mutated version that worked even better than the one nature came up with. That accidental improvement kicked off a race, because a natural enzyme is a good proof of concept, but way too slow and fragile for industrial use. In 2022, a team at UT Austin, led by Hal Alper, took a different approach. Instead of tweaking the enzyme by hand, they built a machine learning model called MUT-Compute and let it scan the enzyme structure for weak points, spots where a different amino acid might make it faster or sturdier. The AI proposed millions of possible combinations. The team narrowed it down to a handful of mutations and built a new enzyme. Fast PETase functional, active, stable, and tolerant. The results were dramatic. At just 50 degrees Celsius, this engineered enzyme degraded plastic about twice as fast as competing enzymes running at 70 degrees Celsius, and it fully broke down an entire plastic cake tray from a supermarket in under 48 hours. The byproducts could then be chemically rebuilt into new, virgin-quality plastic, a real closed loop instead of the downcycling we do today. Now, before this sounds like a silver bullet, there's a catch. These enzymes work best on amorphous plastic, meaning plastic with a loose, disordered molecular structure. But most bottles are crystalline, tightly packed, and much harder to break into. Right now, the plastic usually must be melted down and reshaped before the enzyme can even start working, which adds cost and energy. So, this isn't yet a toss your trash in a bin and it disappears overnight technology. It's a promising early-stage recycling tool, still being optimized for the messy, mixed plastic waste we actually produce. Here's why this research has gotten more urgent, not less. A 2026 study published in Nature Medicine found measurable concentrations of microplastics in human brain tissue, and in people with a documented dementia diagnosis, those concentrations were even higher, with visible buildup in blood vessel walls and immune cells. Separately, a 2024 study in the New England Journal of Medicine found that patients with microplastics detected in their carotid artery plaque had a significantly higher risk of heart attack, stroke, or death over the following 3 years. That was the first major study to connect microplastics to a hard clinical outcome in humans, and researchers are careful to note it shows correlation, not a settled mechanism. This isn't about panic. The science is still catching up, and there's a lot we don't know yet. But it's exactly why enzyme recycling matters. If we can intercept plastic waste before it ever breaks down into the micro and nanoplastics ending up in our bodies, we're not just cleaning up oceans, we're addressing a health question in real time, and the momentum is building. In April 2026, the US government's Advanced Research Projects Agency for Health launched STOMP, a $144 million research campaign specifically aimed at detecting, characterizing, and eventually removing microplastics and nanoplastics from the human body. On the industrial side, enzyme recycling companies are scaling up pilot plants to process real-world plastic waste, not just lab samples, using engineered enzymes descended directly from that original mud puddle bacterium. So the next time you see a plastic bottle, remember, somewhere in a lab, scientists have trained a molecule to see that bottle as food. It's not solved yet. But for the first time, we're not just fighting plastic pollution with landfills and guilt. We're fighting it with biology. If you want to explore more, subscribe for more discoveries like this.